Zach. So today you are going to be learning some science because I feel like teaching you some science. So we're going to talk about DNA replication, and the first thing I'm going to explain, I'm going to just assume you know some basic things, and if I assume you knew something that you didn't know, just ask me about it later and I'll explain it. But okay, so the first thing we're going to talk about is that DNA replication is semi-conservative, and what that means is that each of the daughter DNA strands, so you've got your parent DNA strand and then your two daughter DNA strands, so you've got the original and then, you know, the two that you sort of copy from it. So but what happens is pieces of the original parent cell are in the daughter cell, so it's semi-conservative because your original is still there, semi-ly. And today in class, Sederholm was like, they're semi-conservative, so they can't be teabaggers. And we all just sort of were like, but yeah. But here's a little picture that sort of helps explain it. So I'm showing you a picture. So there, the blue at the top, you've got the parent, and then you've got the two daughters, and see they both still have a piece of the parent, and then by the, is that the second generation? Yeah, second generation. You've got these two here that don't have a piece of the original parent cell, but these two here still do, and you have to sort of think of that as ratios. So somewhere in your body right now, maybe, no, yeah, definitely, is the original piece of DNA that made you, which is kind of cool. Alright, so now you've got that. DNA replication is semi-conservative. Now you know that. DNA replication is also, what was the other one? Semi-conservative and discontinuous, right? So, DNA replication is discontinuous, and the reason we say it is discontinuous is because when you have a double helix of DNA, right, so you've got your double helix thing, what happens is it sort of gets unzipped by words. What is it called? Helicase. Helicase sort of pulls the two strands apart, and then they get copied. And it would make sense, like intuitively, for both to just be copied at the same time, but that's not how it works, because DNA runs anti-parallel. So one is going from 5' to 3', and the other one is going from 3' to 5. Whatever. They're anti-parallel. Confusing myself. Anyway, so as they're being pulled apart, one is just being continually copied, replicated, and we call that one the leading strand. And then the other one is being copied in sort of little fragments that we call Okazawa fragments, and that's called the lagging strand because it is always lagging behind because it takes longer. And this is a little picture that sort of shows what I'm talking about. Right? So there's your DNA, your original one up the top there, and then the helicase is the little round thing that's sort of unzipping them, and then the one that one right there, this one, is the leading strand, and the other one is the lagging strand. So there. Now you know some science. So yeah, I hope that was interesting. I thought it was interesting. I mean, I already knew most of that stuff, but I never really thought about the fact that your original sort of piece of DNA is still in there somewhere, which is kind of cool. I don't know. But that was one of my questions today for biochem, so I thought I'd answer it for you. Also, I'm fiddling with my tea because it is doing the whole Berea College it's raining thing, so Holly and I are in the room right now, and we've made tea. Also, I'm in a cute outfit today. I'm wearing, here, I want to show you. Okay, I'm in like a little top, and then like skirt, and then tights. See? Tights! So yeah. That was weird. A lot of movement. Don't have a good enough camera for all this movement. Alright, I miss you. You should make me a video and talk about puppies or something. Bye!